Hey everyone, welcome back. In our last video, we used a tool called Pandas to take some messy API data and turn it into a beautiful readable table. So today we are going to unleash the full power of Pandas. So in bioinformatics, you rarely work with such just sequences, right? So you also work with metadata, which is massive spreadsheets containing patient information, clinical outcomes, gene expression counts, and tissue types. So usually these are saved as CSV, which is also called comma separated values or TSV tab separated values files. So if you try to open a 100,000 row TSV in Excel, your laptop might start smoking. So Pandas handles it in milliseconds. So let's jump into VS Code and see how exactly. So just like in our memory video, let's generate a dummy data set right here in our notebook so you can code along with me. We are going to create a TSV file simulating 1000 patient samples. So for that, we will first have to import the libraries. Right. Then we will add a sentence here. Right. This command will create the file for us. Then we will write to a TSV file manually to simulate real world data. So for that, we will do .tsv write mode as f dot write uh, sample ID, then tissue type. After tissue type, we will have gene expression, and then BRCA mutation. That should do it. These will be our headers. Then for I in range, since we are going to make 1000 samples, so f dot write uh, this will be uh, sample IDs. After that, tissue types I. Then coming outside, we have here also. I believe we will have to add an I sample ID. After that, we will have to close this bracket. Tissue types. After tissue types, we will have, I think, expression levels, right? So yeah, expression levels I, and one more, which is mutation status I. And then we will have a new line, right? So I think that should do it. Sample IDs, tissue types, expression levels, mutation status, and yeah, that is all. But I believe there is an error coming somewhere. So let me check what the error appears to be. I think we do not need this. Yeah, that should do it. Okay, and then finally we will print a statement. Done. We have our data set. Okay, so let's try to run this cell and see what happens. Right, so we have our data set. So now you'll see a new file pop up called the clinical data.tsv over here. Okay, so let's see what it looks like basically. So there you have it. We have about exactly 1000 samples, right, for this data set. And now let's pretend a collaborator just emailed this to you and asked for some analysis. So if we use standard Python, we would have to split every line by tabs, strip the new lines, and it would be a mess. So with Pandas, it is exactly one line of code. So I'll show you how. You have to write DataFrame is equal to pd dot read CSV clinical data.tsv, and we have to tell uh, Pandas that the separator is basically tab separated, right? That is what we did in the file. It's a TSV file, right? So it will be /t, right? Next, we will display that file after reading it so that we can check exactly what it looks like. Right, let's try to run this and look at how clean that is. Pandas automatically figured out the column names, aligned the data, and even gave us an index number on the left. And if this was a CSV, you would just remove the step is equal to T part, and it will just do the same for your CSV file. Now for the fun part, your principal investigator asks how many tumor samples do we have compared to normal samples. You don't need to count them manually. Pandas has a function for that. Right. So what we will do is tissue counts is equal to df uh, tissue type dot value counts, and then we will print it. Tissue types breakdown, and we will print tissue counts. Let's see what happens. Instantly we get the exact counts. But what if we want to isolate specific data? Let's say we only want to look at patients who have a tumor and are positive for the BRCA mutation. So that becomes multiple conditions, right? So what we will do is basically high risk patients is equal to DF. We will utilize the same data frame that we made earlier. Uh, Tissue type, it should be equal to tumor, right? And tissue type is equal to tumor, and data frame. Inside that we have uh, the RCA mutation is equal to capital P positive, right? So yeah, so since P and everything else is in capital, this should do it, but I believe some error is coming, which could be data frame, right? There should be a bracket here as well. That should do it. And then we will just print a simple statement which will tell us uh, found length of high risk patients, and we will say this many high risk patients, and then we will simply display it. Let's see this syntax df condition, right? It's the bread and butter of pandas basically. We literally just ask pandas to keep only the rows where the tissue is tumor and mutation is positive. It created a brand new data frame with just those patients, and as we can see, it has total of 345 rows and 4 columns, right? So finally, what is the average gene expression for these high risk patients? So Pandas is built on top of NumPy, so doing math is incredibly fast as well. So basically what we will do is average expression is equal to high risk patients, and then we will take the GeneX expression column dot mean, right? And then we will simply print it average GeneX expression in high risk patients is uh, average expression, and then we will keep two floating values after that. And yeah, save our filters results to a new CSV to share with the lab. So high risk patients dot to CSV, and we will name the file as high risk filter dot CSV. And one more thing is index is equal to false. And finally, printf statement saying saved filter data to high risk dot CSV. Now let's see what happens. And we have calculated the average, and then with the dot to CSV we have saved our filter dataset as a brand new file ready to be emailed to the team. And here you can see that file, right? So total of 346 patients showed up for that file. So if you're going to master one library in Python for bioinformatics, make it Pandas for sure. We just loaded a dataset, filtered it by multiple biological conditions, performed statistical math, and exported a clean file all in under 10 lines of code. In the next video, we are going to combine our sequence skills with our Pandas skills. We will learn how to load a FASTA file and turn its properties into a Pandas data frame for advanced analysis. As always, the code will be available on GitHub, and if this video helped you in any way, please hit the like and subscribe button. I'll see you in the next video. Thank you.